Prophase one of meiosis one. This is a very important question in the IP examination. So here, there are mainly five stages, five substages in the prophase one of meiosis one. That is leptotin, zygotin, pocketin, diplotin, diakinesis. Now let us start with the leptotin. So here, chromosomes become gradually visible. Chromosomes become gradually visible under light microscope. Chromosomes become gradually visible under light microscope. So that is light microscope. And compaction of chromosomes continues throughout the leptotin. So chromosome gradually visible under a light microscope, and here the compaction of chromosomes continues throughout the leptotin. So this is a nuclear envelope, nuclear envelope, and this one is a cell membrane, right? This is about the leptotin. So after the leptotin, the next stage is the zygotin. Next stage is the zygotin. That is, here leptotin and zygotin are short-lived. Leptotin and zygotin are short-lived. So here very important, in the zygotin what happens? So paired chromosomes, that is homologous chromosomes are formed. So homologous chromosomes means, so the chromosomes which are similar in size, similar in shape, similar in structure is known as homologous chromosomes. Here, so these two chromosomes are paired and known as the homologous chromosomes. Homologous chromosomes. Here, synopsis, that is formation of a protein complex. This is a protein complex, and this protein complex is known as a synaptonymal complex. So a complex is formed, a protein complex is formed between the non-sister chromatids. These two are the non-sister chromatids of homologous chromosomes. So here formation of synaptonymal complex takes place in the zygotin. So zygotin is characterized with the formation of synapsis, that is the formation of synaptonymal complex. The process of formation of synaptonymal complex is known as synapsis. The process of formation of synaptonymal complex is known as synapsis. So here the complex formed by a pair of synapsed homologous chromosomes is called bivalent or tetrad of chromatids. So these are the bivalent chromosomes or tetrad of chromatids. However, this bivalent and chromatid tetrad chromatids are clearly visible in the pocketing stage. In the pocketing stage. So zygotin is marked by the formation of synapsis. Very important. The synapsis is formed between the non-sister chromatids of homologous pair of chromosomes. This is about the zygotin. And next, pocketin. Pocketin. Next stage is the pocketin. So here formation, the bivalent chromosomes or tetrad chromatids clearly visible. So tetrad chromatids, tetrad chromatids clearly visible. And here, so that is process of recombination takes place in this pocketing stage. Process of recombination, nothing but also called crossing over. That means genetic recombination, that is exchange of genetic material takes place. exchange of exchange of genetic material takes place between the non-sister chromatids of homologous chromosomes. Exchange of genetic material between non-sister chromatids of homologous chromosomes. Homologous chromosomes. Right. So here this genetic recombination or process of crossing over is mediated by the recombinant nodules. Presence of recombinant nodules, and the enzyme is known as the recombinase enzyme. Recombinase enzyme. Recombinase enzyme. Right. So the process of recombination is mediated by the presence of recombinant nodules and the enzyme known as recombinase enzyme. And here the synapsis is continued in this pocketing stage also, which is formed in the zygote. So the synapsis is continued here also. So mainly pocketing is characterized with the process of recombination or crossing over. So this crossing over takes place between the non-sister chromatids of homologous chromosomes. And next stage is the diplotin. Next stage is the diplotin. Right. So here in the diplotin, what happened? This is characterized with the dissolution of synaptonymal complex. So dissolution of synaptonymal complex takes place in this stage. So the process of synapsis is formed in zygote, continued in pocketin, and dissolved in diplotin stage. And the tendency of the homologous chromosomes of the bivalents to separate from each other except at the sites of crossover. So here the tendency of the homologous chromosomes, of the bivalents to separate each other, except at the sites of crossover. Except at the sites of crossover, they get separated. So that means here the X-shaped structures called chiasmata clearly visible. X-shaped chiasmata clearly visible in this diplotin stage. And another important point is the in oocytes of some vertebrates, this stage lasts for months or years. Right. So this is one of the PYQ of NEET examination. So from the NEET examination, so without this topic, we cannot imagine the question paper. So this topic is very important in the NEET point of view also. Right. So the tendency of the homologous chromosomes of the bivalents to separate from each other except the sites of crossover, and in oocytes of some vertebrates, so this stage lasts for months or years. And here X-shaped structures are clearly visible, and these X-shaped structures are called as the chiasmata. Right. And the next stage is the diakinesis. So fifth stage is the diakinesis. So this diakinesis is marked with the terminalization of chiasmata takes place. Terminalization of chiasmata takes place. X-shaped structures are gradually disappear, and condensation of chromosomes takes place here. Condensation of chromosomes takes place in this stage. So this is about the prophase 1 of meiosis 1. And after this stage, nuclear envelope Golgi complex, so they are disappear in the prophase, at the late prophase, right? So once look at the slides. So this is about the diplotin and pocketin, right, and zygotin and leptotin.